Transport across plasma membrane. The internal composition of the cell is maintained because the plasma membrane is a selectively permeable structure. The plasma membrane forms a barrier that blocks the free exchange of molecules between the cytoplasm and the external environment of the cell. Most biological molecules are unable to diffuse through the protein-free phospholipid bilayer or synthetic lipid bilayer. Small non-polar molecules such as oxygen and carbon dioxide readily dissolve in synthetic lipid bilayers and therefore diffuse rapidly across them. Small uncharged polar molecules such as water or urea also diffuse across a synthetic lipid bilayer. By contrast, synthetic lipid bilayers are highly impermeable for charged molecules. However, the plasma membrane, unlike synthetic lipid bilayer, allows the passage of various polar molecules such as ions, sugars, amino acids, and others. Special classes of transport proteins like carrier and channel are responsible for transferring such solutes across cell membrane. Thus, the transport across plasma membrane can occur either through the lipid bilayer or through the transport proteins. The characteristics of lipid phase diffusion and protein mediated transport are basically different. Transport across the plasma membrane is of two types: passive transport or active transport. Passive transport. Passive transport occurs along the concentration gradient and without the use of metabolic energy. If a transported substance carries a net charge, its movement is influenced by both its concentration gradient and the membrane potential, the electrical potential across the membrane. The combination of these two forces, called the electrochemical gradient, determines the direction of transport of a charged molecule across a membrane. Thus, an ion diffuses not simply down its concentration gradient but more exactly down its electrochemical gradient. Passive transport may be simple diffusion or facilitated diffusion. During simple diffusion, a molecule simply dissolves in the phospholipid bilayer, diffuses across it. No membrane proteins are involved and the direction of transport is determined simply by the relative concentrations of the molecule inside and outside of the cell. The relative diffusion rate of any substance across a pure phospholipid bilayer is proportional to its concentration gradient across the layer and to its hydrophobicity and size. Facilitated diffusion. Like simple diffusion, facilitated diffusion involves the movement of solutes along the concentration gradient. However, the passage is mediated by transport protein, which are carriers and channels, and is selective in nature. Facilitated diffusion may be carrier proteins or channel proteins mediated. The rate of transport of the molecule across the membrane is far greater in facilitated diffusion as compared to simple diffusion. Facilitated diffusion allows polar and charged molecules such as carbohydrates, amino acids, nucleosides, and ions to cross the plasma membrane.